joining us for a live town hall. We welcome your Q&A, and we also welcome you telling us where you're dialing in from today. So if you want to add into the chat box who you are, where you're from, and where you're dialing in from, we would love to know that. And I'll be reading those out in just a couple of minutes. If you have a question for George, then you can type it into the Q&A box, and we want your help as the audience in rating those questions. We often have too many questions than we can answer live, so we want your help in upvoting the best questions that you want to ask. Also, if you click raise your hand in Zoom, you will be able to ask George the question yourself. So there are no slides. This is a no slide zone today. And I'm very grateful for George for doing this freewheeling town hall format with us. So there's going to be lots of room for Q&A if I shut up and stop asking my questions. So we'll just have to see whether I abide by my own rules or not. So as I said, we're joined today by Professor George Church. George is a professor at MIT and Harvard Medical School. He almost needs no introduction, but it would be rude not to give him an introduction. He's a pioneer in many fields, from reading DNA to writing DNA to editing DNA, and was one of the leaders behind the Human Genome Project originally to sequence the human genome. Today, we're going to be talking about the work that is going on in George's lab around SARS-CoV-2. How could we not? And we'll also be talking about science and society and how the SARS-CoV-2 outbreak is going to change our relationship with biology. Of course, today is a very special day because it's actually April 1st. And if you're a keen SynBioBeta follower, then you will know that we often put out wonderful, humorous April 1st jokes. Today, nobody's feeling that humorous about the current situation, so we don't have an April Fool's joke today, but I did want to just wind back 2 years and remind you about our April Fool's 2 years ago, which was that Trump had appointed Elizabeth Holmes to head the FDA. That's actually one of our all-time best, I think the all-time best performing article on SynBioBeta.com. And the second was last year, which was Mahmoudi raising their $100 million Series A for cell-based agriculture. And George was very kind to give us a fantastic quote for that. So with that said, George Church, SynBioBeta.
Hi.
You're very busy at this time, and we'll get on to why you're busy right now with SARS-CoV-2 and what's happening with the projects in your lab. But before we dive into that, I wanted to start with a Mark Twain quote that says that history doesn't repeat itself, but it often rhymes. And I wondered if we could if we could go back to 102 years ago, the 1918 Spanish flu. What can we learn from history?
Yeah, so, interestingly, in our personal genome project, we have at least one volunteer who survived that as a 3-year-old. So
Wow.
she's living through both of these historically significant pandemics. One thing we learned, if you look at pictures from that time, is everybody in the streets, or many people in the streets, were wearing masks. They weren't just wearing them. It took us longer than it should have in certain parts of the world. Other parts of the world were very quick to adopt. In fact, they were already using masks before COVID-19.
When you travel to Asia, which I know you do frequently, still every airport will change in the future, which I guess brings us on to this idea of rapid diagnostics, which I was going to get onto a little bit later. But as we brought up travel, how do you think things are going to be different?
It's good. Almost all of these things are good, no matter how small their effect. You add on everybody, and it shouldn't just be during the middle of the greatest pandemic in a century. It should be all the time, and it should be cheap enough that we should distribute it. You should have stockpiles of these things at home because they're so inexpensive. So they're maybe a little more precious and more accurate. But we need to have, as time goes by, there'll be a natural Darwinian survival of the tests that work really well and are inexpensive and very sensitive. If you can detect a single viral particle and distinguish it from a closely related coronavirus, so about a third of common colds are caused by coronavirus. You don't want to have that kind of false positive, but as long as you're willing to confirm it with better tests, I don't think it's a gigantic threat. A much bigger threat is not having the tests, which is what unfortunately happened in the United States.
Can you talk a little bit about these rapid diagnostics, and particularly any of the work that you're doing, either on rapid diagnostics through DNA based systems or through antibody systems?
Right. I think you need both. So our lab has 6 different COVID-19 related projects. I've already mentioned a couple of them. Uh, the You need to be able to know both. So antibodies are, there's 2 ways you can think about it. One is antibodies that recognize the antigens on the virus, or synthetic antigens that you've made to recognize the antibodies in your bloods. And you need to be able to recognize either the viral RNA or the viral proteins as one category of assay, and you also need to know whether you're mounting immune response, and it makes a difference which antibodies you have. So what we really want is to repopulate the workforce with seropositive, meaning having antibodies to the virus, virus negative, meaning having undetectable levels of expression. Because if 80% of the people walking around going to the grocery store are talking out their viruses, in other words, just breathing and talking is producing almost as many viruses per second as coughing and sneezing.
Wow.
If people are talking their viruses, you want to have these 2 tests, virus negative, seropositive. Neither of them has to be expensive, and neither of them necessarily involves blood. So the nasal swab is all you need for the to get the most sensitive virus assay. And for serology, you can do it with just the kind of diabetic finger prick, which I've done many times, even though I'm not diabetic. So I think both of those could be home tests. They could be things you're doing anytime, walking down the street even. That's what we need. And there are a few groups that are working this kind of home tests that don't require instrumentation.
Could we get to the point where we do have tests for those common cold coronaviruses? Can we get to the point where we have a universal vaccine that just gets rid of the common cold and flu, and we don't have this,
Well,
economic?
common cold is caused by more than coronavirus. It's also rhinovirus, respiratory syncytial virus, influenza. There are even some non-viral, bacterial and fungal that have similar respiratory symptoms. It would be nice to know all the different, all these that are present in the environment and their drug sensitivities. So obviously viruses are not going to succumb to antibacterials and vice versa. I've been advocating for a bioweather map since before the 2003 coronavirus. As usual, I'm about very ineffective, but I think that this might raise consciousness and move us in that direction. It's far more cost effective to bring down the cost of individualized home tests. It's like pregnancy tests, very commonly used. We take it for granted that they're actually pretty accurate and affordable, but we could make these things even more affordable. There's no fundamental reason why you can't make these tests for pennies a test. You don't even have postage. You pick up hundreds of them at the drugstore, and then you use them like pH strips.
Great. What's your estimate for what percentage of the US population has already been infected with COVID-19?
Yeah, I don't really know. I mean, I think that it's probably a lot higher than most people would expect, because we're just not testing, and because maybe 80% of them are asymptomatic. I would not be surprised if we get to half of the United States within a few months that have been exposed, and at some point, all those people will be, or 80% of them will be asymptomatic and spreading. And so that's why the point of the mask is to protect others, not really to protect yourself, because most people are going to be endangering others more than they themselves are in danger.
Great. And I do want to do a plug here for Jenscript. I was speaking to them last night from Nanjing, and they're one of the big sponsors of SynBioBeta. And they actually offered to send me some masks personally. They're not the FDA approved masks for hospital use, but they're good for home use or going outside. So if anybody would like masks, just drop me an email and we'll see if we can figure something out. I want to
Well,
go
I think
back
we
to
have a fetish about disposable goods in general. There's masks historically were washable, and
Oh,
and
really?
you can get some really good washable. The N95s are ridiculous. They're made for direct arterial blood into your face. That's you don't need those, especially if your goal is to collect the big droplets that are coming out of your mouth rather than the little particles that are coming in. By the time they dry up, they're hundreds of nanometers in size. While going out, they're hundreds of microns in size.
So presumably there are washable masks out there right now or on the marketplace, are
Well, you
there?
can make one out of t-shirts. Most people have way too many t-shirts. I would recommend it's a lot better than going out without them. If everybody wore some kind of mask that catches the big droplets, plus social distancing and staying at home, this would all end in weeks. It's much more cost effective than vaccines, drugs, and other things. Now we should also do vaccines and drugs, but we need to get this thing under control quickly before it economically bankrupts us. That way we have plenty of money to develop drugs and vaccines and deploy them.
So this is really painful to hear, George, because I think just 2 or 3 days ago, the World Health Organization tweeted against masks. And is that just because of the supply of masks and wanting to make sure that our healthcare workers have them?
I think it was they weren't thinking in every direction. They weren't thinking about, I mean, their job is to communicate, but they weren't thinking about I'm sure that part of it was to try to keep people from hoarding masks. But the problem is, if you tell somebody don't hoard toilet paper, first thing that's going to happen is toilet paper is going to disappear, because they just know that's what everybody's going to be hoarding. And even if they don't understand why it has to be an N95, they'll hoard those things. So I think that was a mistake. The second mistake was instead of saying that this is about protecting you, well, of course it's not going to protect you perfectly. Even the N95s don't protect you perfectly. You don't have a perfect seal around the edge. It's about protecting others, and it's a numbers game. If you can reduce that by 50%, it changes it from 2 million lives to 1 million. That's a big deal. And you put more layers and take your HEPA filters from your vacuum cleaner, and you don't really. There's so many ways to do this. They should have encouraged creativity, and same thing goes for the tests. Instead of saying there's only one test, we're running it at CDC, and then they messed up the PCR primers. They should have said everybody make a test, and we'll let Darwin sort it out. Whoever has the ability to distribute, to get the supply chain, to get people motivated, whatever it takes, and then we'll assess the cost and accuracy. So this Darwinian approach for manufacturing and distribution is way better than letting it Darwinian occur where people are wandering around in grocery stores without masks.
Agreed. This brings us on to the topic of truthiness in science and public policy. You talked before when we did a call to talk about what we we're going to talk about on the show. You talked about the delta between what should have been and what is. Can you just kind of delve a little bit more into that and help us understand what should have been?
Well, this isn't just about the past; it's about the future too. And in the future, it's not just about uninformed politicians. It's a pity that other countries have a lot more degrees in science and engineering than we do in their top level politics. But that wasn't just the problem here. There were people in the CDC, FDA, and WHO which were miscommunicating. And maybe we don't have enough people who have both the communication skills and the out of the box thinking of what the public can do, plus the science knowledge. I mean, that's that rare crossover that we need. But we should have encouraged testing rather than discouraged than trying to centralize it and control it. That could have been done. Ideally, even further back, we would have a proactive testing which is blind to the threat, whatever, just categorizing every virus, bacterium, and fungus. And again, we could be doing that forward. So the one thing that we should have been doing more testing earlier, even before the crisis. We should have been informed by China. The instant that China started spreading, we should have said, let's get our act together because it's going to come over here. So we had more warning than they did, but they got rid of it faster than we did. So that's, even though it had no warning. So that's telling us something about the future. We don't have to go back to 1918 to learn these lessons. And the masks we should have done faster. The vaccines we couldn't have done much faster. I mean, vaccines are a great success story for synthetic biology, but in a curious way. I mean, we can now almost instantly go from patient 0 or somebody around them to a sequence, to a synthetic vaccine, to animal testing in a few days in principle. Okay, but that doesn't help because it still takes 18 months to do scale up and testing. And even for the seasonal flu, which is a very special case, we have much more confidence in its safety. It still takes 6 months to go from, the virus ready to scale up and the scale up and distribution. Now, one of the solutions from that synthetic biologists have pursued in the past is food-based drugs, vaccines. So if that's an interesting possibility where a lot of the expansion could occur in a distributed manner. But you can see how that wouldn't be an immediate easy sell for FDA.
So dig into that a little bit more. What these are viruses that don't have any of the payload inside of them. How would they be distributed in food?
Well, they don't have to be viruses. Many vaccines are either subunit vaccines or even peptides. In fact, it may turn out that there are 3 kinds of antibodies and cell-mediated immunities that can occur. You can have immune response, which is neutralizing. You can have various components which do nothing, and then you can have some that are protective, enhance the pathogen. So antibodies that help the pathogen go through the FC receptor pathway, or otherwise help the pathogen get intracellular. Anyway, so it's helpful to be able to craft the vaccine, but once you've decided how to make it, what adjuvant you need and so forth, then you package it into something that will survive oral delivery and cross either go mucosal immunity or cross the intestinal blood barrier and get and act as if you got it injected, but you didn't. And so vaccines there's a much wider therapeutic range between effective and toxic, compared to a lot of drugs like warfarin is a very narrow therapeutic range. This is quite broad. You can get, as long as it's not alive, you can actually take in quite a bit of an antigen.
Great. We have a few questions on vaccines and PCR. Ian Hayden, if you want to unmute yourself, I'd love for you to ask George your question directly. We have a question from Hannah Jandar. Will the RT-PCR test remain the gold standard diagnostic test throughout the course of this pandemic?
I think it's hard to say. There is a 5-minute isothermal amplification that Abbott has brought out, and there's already tens of thousands of the Abbott devices distributed throughout the landscape, throughout hospitals. So that one looks, could be promising. There are these home test kits, which are also typically isothermal and fast. Mammoth Biosciences did a nice thing where they published the full protocol. Instead of being possessive, they published it as an open source reveal on their 30-minute non-instrumented test. So I think that the 5 minutes doesn't really require an instrument necessarily. What would be nice, and in fact, one of the things we're interested in is developing assays where you don't need to purify the RNA. You simply have to denature the proteins away and go. If you can go straight into an isothermal high temperature reaction, that's an interesting option. There are chemical amplification methods that have been that could be used. So I would not A gold standard may mean that you go to it, once a month to make sure your new things are working well. Maybe it'll be a gold standard in that sense, but it doesn't mean that that's going to be what we test 350 million Americans and maybe billions of people worldwide.
Can we dig into a little bit more of the way that the Abbott diagnostic works? I was very excited to see that 5-minute test just last week. Can you tell us how it works?
I actually don't know in detail. My impression is that it's isothermal, and there are a number of competing isothermal methods. I know from some of my companies that are involved in manufacturing the enzymes, like Alphazymes, for example, and New England Biologic, NEB, that they are. It's desirable if you can keep the number of enzymes down, and some of these isothermals involve like 6 different enzymes. So I think there's a lot of opportunity for innovation here, and this is pretty fast innovation because unlike a drug or a vaccine, you wouldn't want to necessarily just self-administer this because if you have a bad reaction, it could set back you and your research. But with diagnostics, there's so many checks and balances, and it's really harmless while you're working things out. So I think this could be very rapid and immediately impactful.
Great. We have another couple of questions on diagnostics before we dive into vaccines. Roman Herala is in Slovenia. Initial low number of COVID-19 tests was disappointing, seemingly lower than a number that could be generated by a good academic lab. Why do you think the potential of academic research has not been used to a full extent in the USA? And is that changing now? And then somebody else asking, what are the bottlenecks to home-based PCR testing, do you think?
Well, I think the answer to both is manufacturing, and especially manufacturing with high quality. But I think we need to be very careful not to let the perfect be the enemy of the pretty good, especially for tests, because you can always do confirmatory tests. But we are severely underdiagnosed, both before this crisis and certainly during this crisis. I think the academic throughput might be overstated. A lot of it is frustrating when you're in an academic lab and you got a bunch of PCR machines. You say, why don't we just test the whole lab? But it's very different from being able for the academics being able to service an entire country or globally.
Do you think environmental sequencing is going to become more important? Do you think we'll constantly be searching the environment as if it's a food pathogen before it actually becomes a problem?
I would hope so. I mean, I think some people are pathologically phobic of too much information. But those of us that have dealt with a lot of information know that it's better to have it, and then you develop better algorithms and so forth. I mean, I think people panic a lot more when they don't know what's going on than if they have a slightly noisy test that has a known false positive, false negative rate. However hard that is to communicate, it's better than what we have right now.
Great. We have a lot of people dialing in from all around the world. We actually have almost 300 people joining us live right now. So I'm not going to go through the list and welcome everybody, but do appreciate you all joining. If you have questions, type them into the Q&A box. We've got about 16 right now, and we are upvoting the best ones, and I'll take the best ones. And if you want to ask George your question live, then just raise your hand and I'll invite you to join us. Ian Hayden, if you could just give a little background to who you are and what your question is, and then give us the exciting first-person insight into your question.
Yeah, I guess I'll do the second part first there. So, hey, my name is Ian. I live up in Seattle. I was just admitted into the Moderna vaccine trial as a patient. And so my question is, George, what are your thoughts on the Moderna trial, on their mRNA vaccine platform? And then to follow up, are you affiliated with Moderna at all?
Yeah, so Moderna is one of the few companies I'm not affiliated with. I am good friends with some of the founders, but that's it. I first of all, thank you for your service. You are our front lines of volunteers. I'm a volunteer myself on other things, so I'm very supportive of that. The Moderna, I think the Moderna has solid technology, and hopefully. Fortunately, coronaviruses in general are not particularly difficult vaccine targets. I've heard there's up to 40 vaccines in various stages of development. but in addition, we're so lucky that it's not something like HIV, TB, or malaria. Those have been historically very difficult. This is more like the, mumps, measles, smallpox, chickenpox kind of category, which are relatively easy to get safe vaccines for, safe and effective. So hopefully you will be seropositive, virus negative very soon.
Great.
I'm looking
Thank
forward
you.
to it.
Thank you, Ian. And Ian's also one of our fantastic writers at SynBioBeta, so very valuable contributions there, and great to have your personal insight, Ian. So thanks for sharing that with everybody. Jorbin Liang from Yale, what is your opinion on herd immunity? What do you consider the most promising SynBio platform to accelerate vaccine production from the current 18 months to the future 3 to 6 months?
Well, right. So when you're very early in a pandemic, if you can circle each individual, get everybody they exposed and everybody that might be those, the whole community around each case, we're well past that point. We're almost, we will soon be at the point where so many people will have been exposed to it that the vaccine will be kind of mop-up work, but it is extremely important mock-up work. And I think that herd immunity is a nice goal. It's not an excuse for some of the people in the herd taking advantage of the herd immunity. Really, it should be as close to 100% as we can get, even though we know we can get away with less than that. I don't want to give a number so everybody say, oh, well, if you only need 65%, I'll be the lucky one that just hangs back and benefits from the herd. It doesn't work that way.
Isaac Lakhan, do you want to say your question live?
Yeah, this is Isaac. I'm a PhD candidate. Good to see you again, George. My question was, the response to the pandemic has really restructured in some ways the biotech community and brought them together, particularly from like an innovation collaboration standpoint. And I wanted to know, in your opinion, what are the biggest lessons the biotech community should take away, especially from an innovation and collaboration standpoint, from this pandemic response? What should we keep from this response, and what should we change going forward?
Yeah, thanks for bringing that up. I think it's one of the most, there are many optimistic and even possibly beneficial aspects of this otherwise unfortunate event. I think one of them might be, finally, we're learning our lesson about surveillance and sharing. It's very hard. This is something that's very hard or very dangerous to put into silos the way we do so much of biomedical research in these silos. So I don't think we got it right for SARS 2003 or for MERS or for Ebola or for a variety of previous where people would not send out samples. In some cases, they would hoard their innovations. I think this is the opposite. It's really wonderful to see this level of sharing. And also we're finally getting to the point where people can change habits. This is very, very hard to do behavioral modification, even if it's in your best interest. It was hard to get people to stop smoking, to wear seatbelts. In this case, to wear masks. You still get, we're transitioning out of the era where we got dirty looks from the very people we're trying to protect. We're trying to protect them with our mask. So I think all of this, to me, is very hopeful for the future if we can just remember it, along with remember that maybe better than any previous example, this is why we have scientists around. This is why we have the NIH and equivalents that are around the world. We don't want to just make up facts, because when you make up facts or even miscommunicate, it can cost millions of lives.
That's a great quote. George, when we were discussing this earlier, said for the first time in a long time, the entire world is becoming science nerds. Can you give a couple of examples of where you're seeing that happening?
In this, certainly becoming embracing science. You'll see a lot of the Twitter links will go to at least to a thoughtful New York Times piece that itself is linked to the primary data. there is no longer, denialism is not the main topic as far as I can tell. It's more, what can we really do? Do masks really work? How do you make your homemade mask? How do you, when are the vaccines arriving? Are they safe? Where can I sign up to be a volunteer? It's much more, I think it's even better than, some of the previous golden eras of science communication, like when we went to the moon, or when we talked about going to Mars, when we had, plastics, which was the key word in the Graduate movie. I think this is better than all of those put together, better than Tang, and this is really the sort of thing that people can relate to. They have trouble relating to preventative medicine, but here it's so in your face. You want to prevent it, even if all you can do is wear a mask. At least you can do that.
There were two fantastic op-eds that came out this week. I think they were both in the Wall Street Journal. One of them might have been in the Atlantic. The first was by Eric Schmidt, the former CEO of Google, and he talked about a wonderful boom. He talked about the synthetic biology boom that will come because of this, but he also talked about the online education boom that's going to come from this. He said if all education goes online over the next few months, which we can see, certainly it's looking like it's going to, suddenly your school district boundary of where you live isn't a factor in you getting access to world-class education. That, literally, George, you could be teaching high schoolers a introduction to synthetic biology. How do you think our online education systems or our education systems are going to come out of this, and what are some of the positive things that you see around that?
Well, this is something that I've been advocating by teaching online since the 1990s. I was one of the first of the open courseware, later the EduX and various online courses. We've had the technologies since at least 1999 to do pretty good, and it has a lot of advantages. Is that you can divide up teachers into those that are the best lecturers, the best at answering questions, which are not necessarily the best lecturers, they're the best at developing lecture materials for the lecturer to deliver, and then the best for coming up with lab or discussion exercises. And so you need each of these, but you don't need to have every professor becoming the best at each of those, or even adequate in each of those. So I think I'm very much in favor of distance learning, and there's about 1,000 hours of me up on the web so far, if you really want to get an overdose.
Fantastic. Yeah,
so
and I think
I totally agree. This is revolutionary, and moving our bodies around is not only bad for our health, it's also bad for the planet. And so it'd be nice to be able to do some new things. It's also nice that we're being a little less sensitive to eye contact. I find that I'm doing 10 to 20 Zoom, Skype, Google Hangouts per day, and people are getting more tolerant about which way your eyes are looking, and everybody can have their own teleprompter. That's kind of nice. I think that it's going to open our minds. It's always good when you get blasted out of your rut and think about things orthogonally.
Well, I got a glimpse of the future yesterday. I bought an Oculus Quest a couple of months ago, and it's a VR headset, doesn't have a cable, everything's self-contained. I'm going to be encouraging all the SynBioBetans to purchase one. If you want to purchase one, drop me an email, john.cumbers@synbiobeta.com, and I'll send you a discount code to get one. We're thinking of doing a lot more VR events, and tomorrow morning at 8 a.m., we have the founder of Nanome, Steve McGlosky, who's going to be joining us live, along with Rob Reinhardt, who's going to be showing up the spike protein, Robert Scoble, the VR guru, and also Phil Rosedale, who was the founder of Second Life. All five of us are going to be in the Nanome VR environment, and we welcome you to join us live tomorrow at 8 a.m. And we're going to be looking at the spike protein, turning on and off the different parts, and showing how you can actually design drugs in a three-dimensional world. Yesterday was the first time that I'd actually been in a virtual meeting with all five of us standing around looking at this molecule. And we can all move around, we can all bring up different slides, we can all turn on and off and open and close different parts of the molecule. It was amazing. Completely different experience to the Zooms that we've all been on. So there's some very, very exciting things to come in the VR world as well.
Yeah, I agree. I'm as a crystallographer, I was exposed to this three-dimensional imaging in the mid 1970s, but it's come such a long way since then. And I think one of the big payoffs for science communication is indeed for visualizing molecules and cells in 3D.
So the second op-ed that I wanted to mention was by Walter Isaacson. I don't know if you came across that one this week, did you?
I know Walter well. I have not seen that yet, so I will look for it immediately.
Of course, Walter Isaacson, the biographer of Steve Jobs and many other amazing people. But Walter said that this, one of the positive things to come from this pandemic is that the world is watching biology, and that this is going to usher in faster than ever this century of biology, which many of us insiders have been talking about for a decade or more. Now Walter Isaacson is predicting that. So I think we put it in the newsletter just last week, but if not.
And I agree, and it's not like we're just passive beneficiaries of this. I've been impressed by almost everybody has dropped whatever they're working on and figured out the shortest path between what they're doing and what they could contribute to COVID-19. And it's really remarkable. Some of them you would think from their past would not be close, but then you listen to what they're doing, and they're genuinely trying very hard and coming up with good ideas. So I think that once all the dust settles, and maybe there'll be some documentaries and books written, people will see just how each scientist did what and each citizen for that matter became an amateur scientist to evaluate what's coming in through the media, which prior to this was, I think, noisier than it is. Each day it gets less and less noisy. There's less and less. It's easier and easier to find the real facts about what's going on.
George, if I'm somebody who normally works in a restaurant or a bar, or I'm a personal trainer, I train in a gym and that's closed and the bar's closed and the restaurant's closed, and I'm looking at this and I'm not feeling good right now. I'm looking out at a very scary world. What advice would you give to somebody who's watching this at home and thinking, could I get into biotechnology? Could I learn these skills that these people are talking about protein structures, about PCR tests? Any advice that you'd give that would give hope to somebody looking in on this and thinking about retraining and re-educating themselves around a career in biotechnology?
Yeah, I mean, I think you've pretty much already said it, but every revolution that will displace workers. The Luddites were trying to react to the first industrial revolution and just very minor automation. And it's rare. And the problem is you usually don't have the opportunity to both provide for your family and get retrained. But here's an incredibly good opportunity. And boy, everybody who doesn't take at least a little bit of opportunity to retrain, at a minimum, learning enough so that you can protect your family during the crisis, learning what constitutes good hygiene when you're talking about viruses, but going far beyond this, a great time to learn computer programming, whether it's biotech computer programming or not. It's a good time to learn how to tell the difference between facts and stuff that's made up, or stuff that's miscommunicated. So yeah, I think I don't think we need to worry about a glut on the market, having too many skilled people, because biology is growing exponentially, and it's quite possible that almost everything that's currently manufactured without biology is going to be manufactured with it. So now's a great time to get retrained, even if we're very lucky and everybody wears masks and stays inside. You've got at least 3 months to retrain, so do it. Start today. Hopefully you're starting with this conversation.
I love it. Excellent. Great advice. If you want to ask your question live, we have almost 30 questions. So rank them to the top. Stick your hand up if you want to ask it live, and I'll call on you. Carl Schmieder in New York, if you want to introduce yourself. My co-author of What's Your Biostrategy. Carl, go for it.
Hey, John. Hey, George. Thank you so much for doing this. I don't remember the exact question I put down because it's kind of gone up and down on the list of questions, but I'm curious if you see how we can have some ideas in terms of, you actually just mentioned this a little bit, but maybe go into a little bit more detail in terms of how the economy, we can accelerate the rebuilding of the economy with biology and move forward from where we are right now.
Yeah, well, I don't want to sugarcoat it. I think we can destabilize the stock market and our economy long term by saying things are going to be better than they are. So I think it's much better to be slightly pessimistic, plan for the worst, and hope for the best, rather than vice versa. I think there is going to be an economic problem with this many people losing their jobs. Some companies are going under. Now, some of that will give both the employees and the employers a chance to rethink what they're doing, but it's got to be some price to pay. Thankfully, this is not like the housing market, which was basically self-inflicted, but this was partially self-inflicted. In the same case, neither one of them were we putting in the preventative mechanisms. We're not giving enough attention. So hopefully we'll, part of the bioeconomy going forward will be helping this never happen again. I think that people that live through things like World War I, World War II, 1918 flu, have life lessons that they just never forget. But in terms of bioeconomy, I do think that it is much more efficient. We have exponentials where we've improved costs by a factor of 10 million fold. You just don't see that in the steel industry. So if we can expand that into all of these industries, some of them will be exponentially improved and some won't.
Excellent. Thank you, Carl. Zal Bilimoria at Refactor Capital, you're live.
Hey, everyone. George, thanks for taking the time here today. You mentioned that your lab is working on 6 different COVID-19 related projects. In the beginning, you were talking about simple, cheap, virus negative tests, et cetera. What are the 6 tests, 6 projects you're working on, and how can we support them?
Yeah, so, well, they range from just raising consciousness about masks is the simplest and possibly most effective. Neutralizing antibodies. We haven't talked that much about therapeutics, but there you can just use modeling from the SARS 2003 neutralizing antibodies. And this is kind of like getting immune serum from somebody that's recovering, except more manufacturable. We have a test system where we can engineer a human lung, small version where you can do high throughput screening or testing of drugs for safety and efficacy. We have a better, I think diagnostics like masks are one of the things that could impact quickly. And so looking for ways that we can, right now there's a shortage of just RNA prep kits, but there are methods that don't use those RNA prep kits, that don't use some of the precious resources. So working around those. I just working now with Zymergen, and they have a new drug candidate that is one of the first ones that's not just repurposing a drug from another virus. So I think that's very promising. There are a couple others, but that gives you a flavor of what we're doing.
Fantastic. Thank you, Zal. Eugene Joseph, you're live.
Hi, George. Sorry, this is Arun Manoharan from Prime Discoveries. I had a question. So we see that the virus is changing over time. So I want to know if there is a chance for the virus to turn into something more dangerous strain, and how will we keep track of this?
So again, I don't want to reassure. I try to reassure as little as possible, but to think out of the box about things that we could do. In this particular case, this virus not only is a fairly good vaccine target, is well behaved in terms of antigens, but it also is well behaved in terms of mutagenicity. And that's part of the reason it's different from HIV. HIV has a mutation rate that gets up into the 1% range, or single-digit percents. This is has a proofreading polymerase, and so it tends to be more stable, as you would need, since this is the longest RNA genome of RNA viruses. That said, all it takes is one or two mutations to make a radical change in pathogenesis, both for viruses and bacteria. So we should not, and these things come in waves. For example, the second wave of the 1918 flu was far worse than the first one in terms of spreading and killing young people. So I think we need to keep our finger on the pulse. We don't want to just have these simple assays that we want to also monitor the change in the virus and look for patient zero with the new version of the virus. Hopefully we won't just take our masks off and burn them all the instant we've gotten through wave number one.
Great, thank you. We have over 30 questions, and if you want to vote the questions up that you want me to ask, then please do. And if you want to raise your hand, if you want to ask your question live, then please do, and we'll call on you live. I have a question about the 1918 flu, George. Excuse my ignorance. I don't know, how did that end? Did it just peter out? And then also, was there a vaccine created for that strain of the flu? And I'm particularly interested in the person that you said you have in the Personal Genomes Project. She must be, I guess, 105 if I did my math correctly. And so presumably she has antibodies against the 1918 flu.
Well, so I don't know the answer to whether the vaccine status for the 1918 flu. It did, as I understood it, it did peter out in that many people got exposed. That's not the ideal way to deal with this. I'm not sure that people would still have antibodies after 100 years. There are some amazingly long-lived B memory cells, but this would be kind of pushing that limit a bit. You know, with many vaccines, for example, you're encouraged to go back and get them every 15 years or so. It doesn't mean you have zero memory. It just means you don't have an inadequate amount to resist.
Very interesting. So and just for a quick immunology 101, these memory cells are fascinating cells. They're kind of like a library that you have of things that your immune system has seen. And then the memory cells pass on, become, is it plasma cells? Is that where the scale up happens of the antibody?
That's on the scale, yeah. They turn from the memory cells typically just have a few antigens on their surface, and then they're re-stimulated, and they will proliferate and become secretion factories of the secreted form. You also have, you know, cell-mediated, T-cell mediated immune functions as well.
I'm curious, I'm learning so much about viruses and the immune system. I've kind of avoided it because it's so complicated and difficult with many different terms of these different types of cells and these different processes that go on. Can you give us a scale of how complicated the human immune system is as a system that we talk about? I'd say, you know, maybe the brain and the neuronal system is probably the most complex that we don't understand. What are the other big systems in biology that that are out there that are kind of difficult to comprehend, and where is the immune system on that ranking?
Well, the good news is it's much easier to comprehend now that we have more facts about each of these systems. When I first learned immunology in the 70s, there were lots of factors with lots of contradictory and multiple names. Some of them turned out to not be real. And so that was a frustrating time. Now we have a great deal more information, but it's easy to oversimplify these things. You know, I tried to give you a little bit of flavor of the complexity by talking about just within the humoral response itself. There's many different body positions. You know, the gut humoral system is different from lymph nodes, is different from blood. And you can have neutralizing and non-productive immune components and things that enhance the pathogen, unfortunately. You can have autoimmune components. So the complication is not something you measure in terms of the number of cells or the number of synapses in the case of the neuron. It's also you know, how complicated it is to engineer. So as time goes by, engineering is nice relative to science in that you only have to use a subset of knowable information in order to engineer. You can very often engineer with vast ignorance, as was the case for the first vaccine, the smallpox. We didn't know anything about virology or immunology, and it still was possible to engineer a very effective vaccine. So we need to be careful not to let the sort of encyclopedic knowledge and the lack of complete comprehensive descriptions stop us from doing engineering. What the important thing is doing very good testing, safety and efficacy testing to support the FDA, the EPA, USDA, the kind of government agencies that help us stay safe.
Great, excellent. Kira, you're live.
Hi, George. Nice to see you again. Thank you for doing this. I have a question about how this pandemic is affecting your work with making humans multivirus resistant through DNA recoding, as we talked about last year.
Yeah, thanks for putting a plug for that project. You know, we have a lot of projects that that we've mentioned in this conversation that would have been nice if we had them already. The goal there is not so much not immediately to make humans, but to make human cells both for manufacturing and for transplantation. But if you get good enough at making human cells, you can, in principle, make huge fractions of the stem cells in your body resistant to viruses. And that's done by changing the genetic code sufficiently, and it could be as little as 1 or 2 codons, so that you now become resistant to all viruses, including viruses you've never seen before. And this is a perfect example of a virus we had never seen before. We've seen relatives, but that's the point, is we were ill prepared. But we have shown that you can make cells, so far industrial microorganisms, that are resistant to almost all viruses, and hopefully soon we'll show that is a general plan for others. So this is something that probably won't get accelerated by preventative medicine, since we're not particularly good at preventative medicine. It will go in through agricultural and industrial applications, and then into transplantation and stem cell transplants and cell therapies. And then finally, after that, we might get into a preventative world. So it has to go in a particular order, just because of the way we humans think about risks and prevention.
Great, excellent question. Thank you, Kira. Stephen Hubbard, Zymogen, you're live.
Hi there. So my question was, I imagine that one really good way that we could deal with this virus is by having a very high level of testing for the virus and for serum immunity. It makes me think that we should push for some high level of testing before anyone really returns to normalcy. And I'm trying to figure out what that percentage of people who need to be tested for return to normalcy should be. I think 50% of all the humans getting tested is a good idea, but that's arbitrary. Do you have any recommendations for how we should choose that number?
Well, my recommendation is 100%, and rather than making that expensive, we should bring the price down. Just like we brought the price of sequencing down 10 million fold, you can bring the price of testing down into the pennies per test. I'm not saying how fast you can do that, but with the kind of commitment we have right now, almost everybody is now working on COVID-19. I mean, we should be able to bring that price down. The problem in the past is getting people's attention. So to say, here is something we should bring the price and cost down. And also there are less forces aligned against cost reduction. There are occasionally such forces where a company will say, I don't want to bring the price down, right? Because the profit is related to the fraction that you're skimming off. I think that will happen. I think there's a really good chance, and we should push for it. Both for you want serum positive, virus negative, so either viral RNA or antigens. And
what do you think is the right percentage of people who should get these tests?
Well, in some countries, they're actually requiring that you test before you go back to work.
Why aren't we that country?
We are the land of the free, and we are very libertarian by nature, and I think that's usually a very, very good thing. In this particular case, a slightly more a society that's more focused on others than on oneself. You know, the mask to protect others rather than worrying about whether it protects you. The following rules in order to protect the public health rather than. But I think we'll get over it. I think we are getting over it very quickly. People, you know, it's amazing how the threat of your friends and colleagues and family dying, even if it's only 1%, only, it is enough to get your attention. And I think everybody's cooperating now. And I really look forward to everybody getting tested before they go back to the workplace.
Thank you.
I mean, right now, there are a lot of people in the workplace because they're essential, right? But it would be nice if even they got tested.
Thank you very much, Stephen. Great question. We're running out of time. We only have 2 minutes left, so I'm going to ask a couple of very short questions. And sorry we didn't get to all of these. I hope that you've enjoyed this, George, and I hope that we can have you back again. It's the most popular event that we've ever done. So thank you so much.
I'd be happy to come back. Yeah.
Fantastic. So the couple of quick questions. China, threat or opportunity?
Well, actually, I do a lot of work in China, and now Zooming and Skyping to China, and it is very refreshing to see our possible future. Now, there may be a little bit more disciplined. They may have more people in power that have degrees in science and engineering, but you know, when I have teleconferences with them, they're starting to take off their masks and they're starting to go to restaurants, and it's returning to normal. And are they a threat? I mean, well, if this is the era of biology and it is the era, and this depends on people taking science seriously, I think they do have a slight competitive advantage. That said, most of the real big breakthroughs in synthetic biology and DNA sequencing have come out historically and are still coming out from the United States. I don't think we should rest on our laurels. No, I think we shouldn't be so xenophobic either.
Agreed. Have you ever thought of running for office?
Well, thinking is different from doing. I think I'm more effective doing what I'm doing right now. I am very I have helped a number of scientists that wanted to get into politics. I'm very supportive of it. You know, I've done minor things like the March for Science rally with 50,000 people, but I think I think you can do a lot more behind the scenes than you can if you spend all your time shaking hands and going through rituals.
And the final question, how many years do you think COVID-19 has delayed humanity's expansion into space?
Well, oddly, it might actually accelerate it. So another one of my pet projects that hasn't really taken off yet is building space colonies on Earth. These could be anywhere from single person to whole towns, but the point is it's scalable like a trailer park, except hermetically sealed trailers. And the idea is, we shouldn't be testing space colonies first time on Moon or Mars, because if something goes wrong, it's a very long way to the nearest hospital. What we need to do is test it on Earth, and to test, you know, the psychosocial components, the microbiome, the full recycling. We still don't have any really good example of full recycling with humans. We have it with other plants and animals. So anyway, so this biosphere, biobox idea is something that the people in it would be immune to all diseases. They would have zero commute. They'd have the virtual reality jobs. In fact, we might even see a decrease in many infectious diseases during this time of COVID-19. To be an interesting experiment that would be very hard to have done with IRB approval, but it'll just be interesting to see which diseases go down in what parts of the world as a consequence. Like it was interesting to see how many highway deaths were saved when we had various gas and oil crises because
Fantastic.
of the reduction of the speed limits.
George Church, it's been an honor to speak with you today on SynBioBeta Live. I'd like us all to collectively give George a virtual round of applause. Everybody sitting at home. And if you want to send a message to George and thank you him personally, type it into the chat box now, and we'll keep the line open for a few minutes longer than we normally do. And then we'll send all these comments to George for him to look at over the weekend. So George, thank you. This has been fantastic, amazing. Keep doing the amazing work that you're doing. Keep inspiring multiple generations, particularly at this time when everybody's working from home. I hope that we all show this interview to our kids in the future. Thank you.
I just want to thank everybody who is doing their part. It's just amazing to watch people really effectively, not just make believe, moving in this this direction and volunteering, doing whatever they can, whatever your skill set is, just offering your body if nothing else. So it's really very inspiring to me. Thank you.
Absolutely. Thank you. Enjoy the rest of your day on Zoom as well.
Yeah, take care.
Okay. Bye, everyone. Yeah. And Kevin, maybe we can just keep the webinar going for a few more minutes. So if people do want to send a message of thanks to George Church, what an amazing guy and what a fantastic opportunity to ask him so many great questions. We still had 33 questions that we didn't get to. So I apologize for all those people that we didn't get to today. Clearly, George indicated that he'd like to come back on SynBioBeta Live, and we'd love to have him back, of course. As a reminder, we have Nanome tomorrow at 8 a.m., and that's going to be something very special as well. We did a demo of it yesterday, as I described. And also, we have Jim Collins on Friday. And if you don't know Jim, he's also an MIT professor and a trailblazer in synthetic biology. George has 6 projects that he's working on for COVID-19. I think Jim also has, I think Jim has 5. So a lot of COVID-19 projects that we're going to be talking about. And then next week, we just announced, or we will just be announcing today, we're going to have the Chief Medical Officer of Illumina joining us. That's on Wednesday morning, I believe. And on Monday morning, we have Drew Endy. And on Tuesday morning, we have a whole show dedicated to brain-computer interfaces. Yes, you get a little bit of reprieve from COVID-19. Maybe. I'm sure we'll be able to fit it in somewhere. That's with Matt Angle. And Matt is the CEO and the founder of Paradromics. And Paradromics is a brain-computer interface company. So they've got this high throughput way to stick probes into brains and read signals in and out. That's going to be on Tuesday of next week. So we have a lot of really interesting stuff coming up. If you're not subscribed to the SynBioBeta newsletter, then you can go to synbiobeta.com and subscribe. And you can follow us on Twitter and LinkedIn. And today's show was broadcast live on YouTube. So if you're joining us from YouTube, then hi to you too. You can also join via Zoom, and you can find out the Zoom login information if you sign up to our newsletter or go to our website. So thank you all. It's been wonderful to chat with George today and wonderful to get so many great questions. So we appreciate all of you for tuning in, and we'll hopefully see you for the Nanome Town Hall tomorrow at 8 a.m. Have a great day. Cheers. Bye-bye now.